Okay, so in this video, I'm going to cover the need for gas exchange and surface area to volume ratio, and then gas exchange in leaves, fish and insects. There'll be a separate video for gas exchange in mammals. So the first thing to think about is what we call the surface area to volume ratio. So if you take a cube, and imagine this cube at the moment is a 1 by 1 by 1 cube, doesn't really matter what the units are. And then we take another cube. And this cube is a 2 by 2 by 2. And then we'll take a third cube, obviously not drawn to scale. And this cube is a 10 by 10 by 10. Now, if we calculate the surface area and volume of these three cubes, then we can look at the relationship between surface area volume ratio and the size of an organism. So the surface area of a cube obviously is 6 times the surface area of each face. So the surface area of each face here is 1 times 1, which is 1. So the surface area is going to be 6 times 1, which is 6. But the volume of this cube is 1 times 1 times 1. So the surface area to volume ratio is 6 to 1. In other words, there's 6 times the surface for every unit of volume. If we look at our 2 by 2 by 2, so each face of the cube has an area of 4. There's 6 faces, so we have a total surface area of 24. The volume is 2 by 2 by 2, so that is 8 in total. If we simplify that, that now gives us a 3 to 1 ratio. So when we've increased the size of our cube, our surface area to volume ratio has now dropped. We've only got 3 times the surface area for every unit volume. And if we go to our 10 by 10 by 10, so our surface is 100, so we have 600 surface area, and we have 10 by 10 by 10, which is 1000 in terms of the volume. So we actually now have only 0.6 to 1. In other words, we have less surface area for every volume. This creates organisms a real problem. When you're really small, or if you are designed to be long and thin, okay, so if we looked at this organism over here, for example, which was 1 by 1 by 10, okay, then its surface area would be sufficient to absorb its needs across its surface, absorb the oxygen that it requires, get rid of the carbon dioxide, not a problem. This organism over here, it's got 6 times the surface area to its volume, so therefore it can absorb its requirements across its surface. But once you get to an organism this size has a particular problem because it now cannot take across its surface the amount of oxygen it requires for all its volume of cells. So it's got to have specialized exchange surfaces to allow it to do that, and then specialized transport surfaces to actually transport the substances around, because the diffusion distance would just be too great. So a small organism, not a problem. It can absorb its products across its surface. Big organism, like fish, insects, mammals, they're going to need special exchange surfaces. So the first organism that you looked at was actually a plant. It was a leaf. Here is the leaf structure as you're used to it. Upper epidermis, palisade cells, spongy mesophyll cells, which are irregular with lots of air spaces. And then our lower epidermis with our guard cells and our stomata. And the important thing to notice is that gas exchange is taking place across the stomata. With carbon dioxide going in, it's required for photosynthesis, and oxygen, which is produced in photosynthesis, leaving. And these air spaces with the cells around it, they provide a large surface area for the gas exchange to take place. Next, we look at fish. Now, fish are a little bit more complicated. You should know that fish have gills, and they take in their oxygen and exchange their carbon dioxide across those gills into and out of the water. If you look at the gills in a little bit more detail, they are made of these filaments, and on each of these filaments there are these lamellae. Okay, so the lamellae, which are here, they're going to provide a much larger surface area. So by having lots of filaments plus lots of lamellae, we have a large surface area. And across these lamellae, if you look at them, you have the blood flow. Okay, so you've got the deoxygenated blood coming in, it's then flowing across the surface like this and becoming oxygenated. So they have a good blood supply. Okay, this is really important, as we shall see later, because a good blood supply is crucial for maintaining that concentration gradient, because all of these absorption of gases is all about diffusion. So we've got a large surface area and we've got a good blood supply. You'll notice also that the lamellae are thin. Okay, thin allows a short diffusion distance. So we already have 3 adaptations that our fish have. Large surface area by having lots of filaments and lamellae, good blood supply to maintain a concentration gradient, thin to make a short diffusion distance. But that leaves us with one more crucial one, which is the fact that there is what we call a counter current between the blood and the water. And that's what I want to explain a little bit more now. So if we think about the counter current mechanism, counter current mechanism is very simple. It says that water flows in one direction whilst the blood flows in the opposite direction. And the reason for it is it maintains the concentration gradient. And I'm going to show you why with reference to 2 graphs. So if we imagine, first of all, that we have the graph and we imagine it was not a countercurrent multiplier. Okay, so if it was not a countercurrent multiplier, which we know is wrong, what we would find is our water would be coming in with, say, 100%. Okay, so let's say this is oxygen concentration. Okay, and this is the length along the lamellae. So we're going along our lamellae here. So the oxygen comes in at 100% and the blood is coming in near 0. And obviously there's going to be a concentration gradient because the water has much higher concentration than oxygen. But as it goes down the lamellae, it's going to decrease. And what's going to happen is that the blood is going to increase and they're actually going to meet. And about halfway along the lamellae, you're going to stop having a concentration gradient. So you're doing fine from there. You're getting movement, diffusion, but then you reach there and it just stops. On the other hand, if we have the other way, so we've still got length and we've still got oxygen concentration. Okay, and we think about the water. Okay, so the water's coming in one way and the blood is coming in the other way. So the water, say, is coming in from this direction. It starts at 100% and then it's slowly going to drop. The blood, of course, is coming in at the other direction. So it's coming in from the far end. So we're going to start it over here with 0. And as it goes along, it's going to pick up oxygen. So what you see here is that all the way along the length, all the way, you now have a concentration gradient. Okay, so this is your water in both ones, and this is your blood. And that is the reason for the counter current flow, because it means you've got a concentration gradient all the way along the length. Now, the last animal in this particular video we're looking at is the insect with its tracheal system. So this is when you're looking along the body of an insect. So imagine you have an insect. I'm not going to draw all its legs, etc. You're looking along its body, and what you can see in the abdomen and the thorax are these holes. And these holes are called spiracles. And the beautiful thing about the spiracles is they can open and close. They will open when the CO2 concentration in the tissues increases, because that tells them that the organism needs more oxygen. So gas exchange, gas comes in here, okay, it then goes into the trachea, which we've got over here. And you can see the trachea is held open, okay, by rings to keep it open. We then go down into tracheoles, which are down here, which leads into the tissue. Once again, the same principles apply, because we've got gas exchange system. We have thin walls, which is a short diffusion distance. We've got many tracheoles, which equals a large surface area. Which equals a quicker rate of diffusion. You haven't got a blood supply in this case because they have an open system, but you do have ventilation to maintain the gradient. But there's another neat little thing that's worth knowing about, which is the liquid that's in the bottom of the tracheoles. And I'm going to illustrate that with a diagram underneath. So if we have our trachea coming in, then we have our tracheoles, like so. And then we have our tissue, which is around here. If we have a look inside the tracheoles, what we see is that there is fluid inside them. And gas exchange can only take place across the area where there is no fluid. So here you're going to get oxygen going out, carbon dioxide coming in, and so forth. Okay, only where there's no fluid. However, when the cell is really, really active, okay, so when we have active cells, for example, an insect that's flying, you're going to get anaerobic respiration taking place. That's going to lead to the production of lactic acid, or lactate, it's the same thing. That's going to lead to a low water potential in the tissues, and that leads to water moving out of the tracheoles, which means that you end up with less fluid. Okay, so if we rub out some of our fluid here, oops, we need to do that one. Okay, rub out our fluid there, rub out fluid there, and we'll just put a little bit of fluid in. Okay, so now we've just got a little bit of fluid. Look what we have done. We have now increased the surface for gas exchange. And by doing that, the cells will now get more oxygen, which means that they can do more aerobic respiration. So less fluid means that we now have a larger surface for gas exchange. Which supplies more oxygen for aerobic respiration. So they can switch from anaerobic to aerobic.